Welcome to this explainer on precision nutrition modulators of allergy and immune function. We have four main learning objectives for this session. First step, we need to differentiate innate and adaptive immunity. Next, we will diagram the exact mechanism of GALT activation. Third, we are contrasting IgE-mediated food allergies with functional food intolerances. And fourth, we evaluate the mechanistic impact of ketogenic diets on Th17 cells. To get the most out of these mechanisms, you need a bit of prerequisite knowledge. Specifically, we expect a foundation in basic cellular biology, the fundamentals of macronutrient metabolism, and the anatomy of the gastrointestinal tract. Section 1, Problem Framing and Significance. Think about this. Allergic diseases affect roughly 33% of the general population. This represents a massive portion of people. Atopic allergic disorders classically involve an exaggerated IgE-mediated immune response. You usually see this confirmed via high allergen-specific IgE and blood serum or positive skin tests. Because individual disease progression relies heavily on distinct genetic backgrounds and metabolic states, broad population-wide dietary guidelines consistently fail to address individual dysfunctions. Targeted nutritional interventions offer a framework to correct specific metabolic imbalances at the individual level. When we look at nutritional extremes, they trigger distinct physiological impacts on systemic immune responses. Take malnutrition, for instance. It leads directly to impaired immune function and depleted body protein stores. On the flip side, obesity induces chronic low-grade inflammation. What happens is excess intra-abdominal fat outgrows its blood supply. This causes cellular death and releases dangerous signals into the body. These signals attract macrophages, leading to the secretion of tumor necrosis factor alpha, driving insulin resistance. Section 2, Core Immunological Concepts. Let us define innate immunity. It functions as your body's first line of defense. It integrates physical barriers like skin and gastric acid with nonspecific cellular responses. The primary cellular mediators here include monocytes, macrophages, and dendritic cells. Upon detecting danger signals via toll-like receptors, these cells secrete pro-inflammatory mediators. We are talking about tumor necrosis factor alpha, interleukin-6, and reactive oxygen species. This rapid response requires zero prior exposure to the pathogen. It happens instantly. Then we have adaptive immunity. This system executes antigen-specific T cell and B cell responses, relying heavily on specialized CD4 positive and CD8 positive cells. The adaptive system possesses a unique capacity, generating long-lived memory cells. These cells remain primed for highly targeted protection against specific pathogens or even tumor cells during future exposures. This gives the body long-term biological resilience. Now consider the gut-associated lymphoid tissue, or GALT. It constitutes the largest single collection of immune cells in the human body. GALT manages a delicate biological trade-off. It has to eliminate harmful pathogens while actively maintaining self-tolerance to normal food proteins and commensal microbiome bacteria. Grasping how GALT functions proves essential for evaluating oral tolerance and mucosal immunity. A critical factor altering these immune systems over time is immunosenescence. This is the age-related deterioration of both innate and adaptive immune function. A primary driver here is thymic involution, resulting in fewer naive T cells over time. As human stem cells age, they differentiate preferentially into myeloid lineage cells rather than lymphoid cells. This shift reduces overall T and B cell formation. Physiologically, this correlates strongly with chronic low-grade inflammation, frequently termed inflammaging. Section 3, Mechanisms of Immune Diet Interaction. The sequential mucosal activation pathway originates right in the small intestine. Step 1. Dendritic cells sample commensal antigens from the gut microbiota. Step 2. This sampling initiates the differentiation of regulatory T cells and B cells. Step 3. These newly differentiated cells migrate to localized anatomical structures called Peyer's patches. Step 4. Within these Peyer's patches, additional stimulation yields the production of commensal-specific immunoglobulin A, effectively establishing self-tolerance. Localized commensal bacteria simultaneously inhibit pathogenic microbes by competing for scarce nutrients and inducing specific T and B cell responses. Microbiome dysbiosis completely disrupts this localized homeostasis. Gram-negative bacteria within an imbalanced microbiome release lipopolysaccharides, or LPS, directly from their outer membranes. This LPS degrades the intestinal barrier, increasing overall intestinal permeability. Endotoxins subsequently enter systemic circulation. This creates metabolic endotoxemia and drives pro-inflammatory reactions strongly linked to obesity and diabetes pathogenesis. Managing gut-flora equilibrium proves vital to maintaining barrier integrity. Section 4. Case study on ketogenic diets. To induce physiological ketogenesis, the nutritional intervention requires stringent macronutrient constraints. The diet necessitates severe carbohydrate restriction while mandating 70% of total calories originate from fat. The objective here is forcing increased hepatic production of ketone bodies. Because a strict ketogenic profile proves difficult to maintain for many people, fasting mimicking diets, relying on low calorie and low protein parameters, serve as alternative methods to induce the state of ketogenesis. Studies employing human microbiome transplantations into germ-free mice isolate the direct molecular consequences. Ketone bodies, specifically beta-hydroxybutyrate, alter intestinal cell communication. The localized effect heavily reduces the population of pro-inflammatory Th17 cells within the lamina propria of the gut-associated lymphoid tissue. The data demonstrates a clear, direct mechanistic link between macronutrient modulation, ketone generation, and a suppression of intestinal inflammation. Suppressing these pro-inflammatory pathways mitigates localized tissue damage. Section 5. Common misconceptions and pitfalls. A prevalent clinical error involves confounding food allergies with food intolerances. They are entirely different physiological events. Food allergy represents an immune-mediated hypersensitivity. It requires an IgE-mediated or T-cell-mediated immune response to specific antigens, leading to severe reactions, including anaphylaxis. 90% of food allergies trace back to milk, eggs, peanuts, tree nuts, fish, shellfish, wheat, and soy. Food intolerance, conversely, represents a non-immune pathophysiological process. It constitutes a functional gastrointestinal limitation like a specific enzymatic deficiency, such as lactase absence. While food intolerances affect roughly 20% of the population and often overlap with irritable bowel syndrome, they entirely lack the immune system activation defining an allergy. A second significant pitfall involves relying on standardized micronutrient recommendations to assess immunological endpoints. Current dietary guidelines for vitamin D reflect endpoints strictly related to bone health. The optimal serological levels required to maximize immune function remain an open scientific question. Precision nutrition analysis reveals vast inter-individual differences in vitamin D metabolism based on distinct microbiome profiles and epigenetic variables, rendering generalizing supplementation advice suboptimal for immune restoration. Section 6. Summary and conclusions. Multi-omics predictive modeling translates massive arrays of phenotype data. This incorporates host genetics, continuous glucose monitoring, and precise microbiome sequencing. Machine learning algorithms process these diverse biological inputs to generate highly personalized dietary recommendations. The field is rapidly transitioning away from generalized guidelines toward precision models, aiming to optimize immune function, manage chronic inflammation, and prevent diet-related diseases based entirely on individual, quantifiable biological data. So ask yourself, how will the integration of your unique microbiome data alter your personal nutritional requirements in the coming decade? And if you like this video, hit that like button, and please consider subscribing to my channel.